So that is all I wanted to show at the moment for structured illumination. And then the next topic is what I call pointillism. So it is imaging single molecules. And so in pointillism, the trick is that you want to actually localize single molecules rather than imaging the whole bunch of them. So I would refer to structured illumination, nonlinear structured illumination, or stimulated emission depletion microscopy as methods that do not rely on the fact that nature is sort of separated into molecules. They might use photophysical properties of these molecules. However, they do not need them as single entities. STED doesn't care whether in the middle there's a single molecule or 2 or 3 or 10 or thousands of them. In fact, it would even benefit in a way from a continuous world. Whereas the idea behind particle localization is that ultimately you're always looking at the single molecule entity as a spatial thing, that you're based on the model that there is only a single molecule that gives rise to the detected signal in some way. And this is a bit on the history of this. So actually there was a really nice paper, a general paper by Eric Betzig, I think in 1995, that laid out the concepts, and that if you can separate single molecular entities, you can localize them precisely. You can get high resolution images. And then one approach to separate molecules was now in the group of Christoph Kremer, which is what is called the spectral precision distance microscopy. And the idea is to separate the different molecules or the different entities, let's say, by color. And at that time, this was then done basically by localizing, for example, different genetic loci in the cells and measuring distances between them. That's why it's called precision distance microscopy. So that worked, but the problem, of course, is that an image typically contains more than just 3 or 4 or maybe 5 positions that you measure. So this doesn't get you very far in number of entities that you can look at. A little bit better it is when you're using fluorescence lifetime. So you can then combine maybe color with fluorescence lifetime. You can get possibly to more single entities you can see, but it's not a high number yet. And I think the first time that it was shown to get a really higher way of separating overlapping point spread functions was by using the blinking characteristics. So basically, this will be shown in the next slide. The idea is this: when this was actually some images that show quantum dots, and these quantum dots, as you see, are blinking. And then when you look at such a field of blinking quantum dots, you eventually start to realize, for example, here there are 2 of them that are overlapping, but sometimes you see only one of them. For example, the left one. Sometimes you see the right one, sometimes the left one, sometimes both of them, and sometimes even none of them. So if you look at that, you think like there must be a way to somehow separate them with some mathematical means. Yeah, here's another example. They're almost overlapping. You can nicely see how they are blinking. And it turns out that there is a method. There's actually several methods, and one of them is independent component analysis. And so that's what we use. So you can basically put all the pixels into a statistical analysis, and it allows you to then separate the entities of the things that do the same together, the blinking molecules. And once you have separated them, you can then localize these individual emitted light from a single molecule, or in this case, a single quantum dot, and you can then start to paint an image of these single. So here's an example how this works. And in this case, all the pixels from the snippet were put into this analysis, and the algorithm came up with these individual images showing only one of the 2 molecules. So this isn't surprising at all, because you can see yourself that these are 2 molecules. However, it's interesting that the algorithm was actually able to pull things apart and make it into 2 molecules. So because we didn't tell the algorithm any information even about where each pixel is located or something like this. And the fact that we see 2 separate entities are simply based on the statistics that the brightness changes together basically in this entity. So these are This is one picture that shows how brightness changes together, and another one changed also together. So this is independently blinking. Yeah, this is what independent component analysis allows you to do. So for the expert, it's a bit like principal component analysis, but it does not require orthogonality of the images, because these two images are clearly not orthogonal in the sense as that their product would yield 0. So it gets much more interesting if you look at something like this, where we cannot even see that these might be two molecules. Yet the algorithm is somehow able to separate this into one image here and one image here. And if you localize them, you find that they have a distance of 23 nanometer. And you can ask yourself, how many of these different molecules can we still statistically sort of separate? And at that time, it turned out to be 5 spots that are all overlapping, can be reliably separated, and there were some other additions to that. And you can then, of course, also make a negative control in the sense that you're forcing the algorithm to believe that these are two particles, and it starts separating them. And then what you get is these two results. And as you can clearly see, it didn't separate this spot into two separate spots, but it separated it into a background term and a spot term, which I think is another confirmation that this really worked very well. So this coined us to sort of, in the abstract, to say that we imagine this method to be called Pointillism, because when applied to labeling structures, then you can make a high-resolution image by consisting of individually localized points that may be reconstructed. So then actually, 1 year later, Eric Betzig published a much simpler method that then was a lot more successful, of course, because almost everybody could do it, simply localizing individual molecules and avoiding totally this complication of overlapping images. So the idea is the following. If you have a single point, then its emission leads to a fluffy image. So of course, this fluffy image, you can still calculate where the original emitter was. You can determine, for example, the center of mass or something like this, or rather a fit of this sort of point spread function shape to this blob gives you a very precise, accurate of the position. The problem is, of course, that in reality, you don't normally know whether this is a single point. For example, 2 points close together, because the image of the 2 points would look so similar to the original image of a single point, especially when these 2 points are very close together, that you can actually not really separate it. Well, it depends on signal to noise. Actually, if you look in my thesis, you find an analysis of this situation, which shows you that about to 1/10 of the distance, you can still, when you know it's 2 molecules, you can still localize them reasonably precisely by the right fitting procedure. But in practice, you don't know, is these 2 or 3 or 1 and so on. So now, first message is you can localize a lot more precisely than the width of this thing is. So it's like the cross on top of a mountain is much more accurate than the width of the mountain itself. So localization is much more precise than the resolution limit of. So if we now have a complicated structure, normally we would have lots of fluorophores on it, and they would all blur together, and you wouldn't have a chance to really reconstruct that structure any longer. However, now the idea of Eric Betzig is, what if you are somehow statistically able to see only single molecules? Here it is pretended that you are only seeing a single molecule at the time. And this way we can then localize them as shown by the red spots here. And by then drawing all these positions, eventually if we have enough of them, then the structure starts to emerge again of what we wanted to image. So of course, in reality, maybe you're not seeing a single molecule, but you're seeing several of them. But the chance of collision, especially if the total rate of molecules is low, goes with the square. So the less molecules you're seeing at a time, the square less chance you have that they collide and that it causes an error in the reconstruction. So that's, of course, a very essential insight here. And how can this in practice be done? Well, at the time from Eric Betzig, this was done with photoactivatable molecules. So there was a very new molecule by Patterson in the group of Jennifer Lippincott-Schwartz that was dark, and it could be photoactivated with a blue light. And then Eric Betzig thought, well, if you just activate very, very little, then you would turn only a few of these molecules on. You can image the heck out of them until they're bleached. You get a nice statistic, can localize them, and then you're activating another few of them. And then you do this again and again and again. And Harold Hess and Eric Betzig have done this in their garage, basically, and they were then able to reconstruct an image. Even the first time they did that, it took them more than a night to basically get all that data. But they got their images. Nowadays, this is a little bit different. You are actually doing this with most of the time with molecules that are pushed into a dark state. So you see this here, that you have a singlet ground, singlet excited state, but occasionally they can cross to a triplet state or even to one of these very long-lived dark states. So this is a chemical change, basically an ionic change. So the molecule has either lost an electron or gained an electron. And by putting enough light on it, you can drive molecules quickly into these dark states where they live for several seconds. And then you have enough time. Well, if you put the conditions right, usually lots of thiols in the medium and so on, then you can make everything dark, and only a few molecules eventually sort of drip back into the state where they can be excited to cause fluorescence to happen, and then you see them for a brief moment of time very, very bright. You can localize them, and then they are gone again, either being bleached finally or being again in one of these longer lived dark states. So this method is called direct stochastic optical reconstruction microscopy, so STORM. Pretty much the same time as Eric Betzig published, there was a paper by Zhuawei Wang, and this is very closely related to work of Markus Sauer that basically described these blinking mechanisms before. And and the direct stochastic optical reconstruction microscopy is the method that Markus Sauer invented. The STORM is based also on a 2-wavelength scheme originally. And well, the idea is that you put the molecules in these long-lived dark states, and you can single them out. And again, I want to stress it's about that you're looking at single molecular entities, which allows you to localize them. You can relate, if you want, localization in a way to a nonlinear process. This is actually something that Stefan Hell pointed out in an early publication, that by sort of clumping together the various photons that are emitted from a single molecule, and by knowing that they are emitted from a single molecule, you in a way you're taking your point spread function to the nth power, so to say, and therefore you're narrowing it down. So this is from the publication of Eric Betzig and the original publication, and it shows you nicely this visualization. So on the left hand side you see the individual snippets of such a movie of single molecules. So you see these blobs are usually mostly single molecules. Sometimes they might be colliding. There are algorithms that can throw these out because they're too bright or something like this, but anyway, it happens fairly rarely, so you can also in some way live with the errors that they cause. And on the right-hand side, you see the localizations that are put on a much higher resolved, so finer pixelated localization map, and they're binned down there. And if you then put them all together, eventually you're coming up with an image. And so they are sort of in this image, these stack over time is sort of raining down onto your final image. And when you've, like you see here, when you've acquired enough data, you get a good representation of your biological structure. This shows you an example also from that publication. So this would be a wide field image of these mitochondria, and this is the palm image, so photoactivated localization microscopy. And what you see is these structures, and they look a lot sharper compared to the left side, but to appreciate how good they really are, it's actually fantastic if you overlay them with the electron microscopy image, as they have done, and you see that here. So you can now appreciate that the resolution is really very, very high because you get this perfect agreement of the palm data or reconstructed palm data with the electron micrograph of the same section. So this was also done with Eric Betzik and Harald Hess. Well, I want to make a remark on localization. So it's kind of interesting because I think there's often a confusion and people often, in a way, wrongly use terms like full width half max or sigma or so for so the standard deviation for describing the localization. And I want to show you why I think this is sometimes a bit dubious. This is okay. One way you can think about localization precision is like this. Imagine we had a Gaussian point spread function. Of course, in reality, it is not Gaussian, and in some way, it's even very non-Gaussian because the Gaussian would have sigma, which is very finite, and it turns out in a point spread function, the sigma would actually be infinite. But nevertheless, so what I've shown you here, these flashes of light where the single photon sort of reigning in this point spread function. So you can interpret the imaging as a stochastical process where single photons are sort of randomly reigning onto this probability distribution described by the point spread function. So if you think about it like this, then measure the position of a particle. What you normally do is you get the best estimate of the position of this particle, so in other words the fit, simply by getting a center of mass. So that means you take the mean position of all the individual measurements of the position. So that's what you often do in physics. You have many measurements that are statistically independent. All you do is you take the mean, and of course, as everybody knows, the error of the mean scales with square root of the number of measurements. So basically that would predict that your localization precision is roughly 1 over square root of n being the measurement, or rather sigma over square root of n. So with every new measurement beyond n, you're getting square root of 2 improvement in, well, actually not with every factor of 2 more photons, you get square root of 2 improvement in the sigma. Right. So that's noise. However, it's not quite as simple as that in reality because, for example, the point spread function is not a Gaussian. If you imagine a different situation, like in electron microscopy, for example, where you have a negative contrast, the point spread function would sort of look like this. So a single dark particle would absorb something. And of course, if you tried the method here, the center of mass would actually not work at all, would just give you complete nonsense because most of the times your photons are appearing in completely different places. Nevertheless, you can still fit here. You have to use a maximum likelihood fit with the right photon statistics, can be a Poisson-based model. In the electron microscopy, this is typically so high that even a Gaussian would actually work fine. Which is, by the way, typically also done in light microscopy, even though this is not necessarily optimal. Well, another comment is on pixel size. So if you now have this model in mind, how high should you sample? And the interesting point is here now you can think about that when you have a pixelated detector or a certain integration time while you're doing your confocal scan or something like this, you have these bins, and that means you're losing information about where the photons were actually hitting. So all you get is this photon appeared in pixel 3, but you forgot about was it on the left side or on the right side or in the middle. So in a way, it introduces a second uncertainty about your measurement. And precisely speaking, you can model the pixel, for example, as a rectangular function. You can still calculate the sigma of that rectangular function. And in the sense of the central limit theorem, that everything becomes more Gaussian when you're basically multiplying or rather convolving these effects. You can estimate that the final error you will be getting by adding the variances of the localization precision with the variance of the pixel size and then taking the square root to get the standard deviation. So this would actually tell you that it is worth taking a little bit more pixels than you would normally do when you image. So in imaging, the rule is you have to do Nyquist sampling. This stochastical view tells you, well, sampling a bit higher than Nyquist, if you're not getting extra read noise in this case, is worth it because you get a more precise estimate. And it turns out that there's even some other reasons to do so, which is that your fitting usually doesn't account for the right shape factor of the pixels and so on. That means if you're not very, very careful in correcting all these effects, then you are even getting certain biases in your localization, which you can avoid also by sampling higher. So my recommendation would be to typically sample about 1.5 times higher than the Nyquist limit. You can find more details on this effect also in my PhD thesis from 1999. So choose your pixel size smaller than the Nyquist sampling would require. So now we come to the point that I was trying to mention before. Let's imagine we had some crazy point spread function, which is 2 delta functions. The question is, does that still apply what I said before? And it turns out not quite. Look at this. The first photon hits here. And if you think about it, where could the original object have been? So for our point spread function, let's say the object is here. The point spread function has a delta peak at minus x and plus x. Then we have 2 solutions. The object could either be here or the object could also be here. Both of them are good solutions. So we now measure a second photon, and if the second photon by chance hits the same place as before, well, we haven't gained any additional information. The object could, with the same probability, still be here or here. So you see, even though we have measured 2 photons, we should be at square root of 2 better now. We are not in this particular case. However, in case the third photon hits, and it hits here, then our localization precision, according to the equation, would be something like divided by square root of 3, but in reality it has suddenly been reduced to 0. So before we had an uncertainty either here or here, now we have no uncertainty, it must be here. There's no other explanation to measure what we've measured, i.e. 2 photons here, 1 photon here. So you see that this argument with sigmas really doesn't apply. It's very unrealistic. It only applies to Gaussian point spread function. It doesn't really apply to something like this. So what you really have to do, of course, is a much more detailed analysis with Kramer-Rao bound and so on, and that would then make slightly more correct predictions. So the standard deviation is not an appropriate measure for localization precision. High frequency content is very important when you're fitting. So this is shown by this example here. Yeah, as I said, Kramer-Rao bound, which is based on Fisher matrices, which is information theory, is what you need. Another comment on localizing. So as I said, the point spread function has theoretically an infinite sigma. You can write down the Jing function and you can see what is the sigma you'll obtain in infinity. So therefore, center of mass is really not useful at all. However, in practice, this is still used a lot. And the reason is that you have to anyway apply some sort of thresholding. So that means you have to say that there's noise on the detector everywhere and maybe even background noise from out of focus molecules. So to gate this out, you need to say, I only consider signal to be signal when it is above a certain threshold, like you see here. Now the question is, how do you do the center of mass? And one way is you could count this green area, which is above the threshold, and determine the center of mass of the green area. But there's of course a problem when you do so. Even though this sounds like a good idea, you are suffering from the following problem. When there's just a tiny change in the threshold value or a tiny 1 photon more in this pixel here, suddenly there's a whole new bin coming in and it drags the center of mass to the side. So COM stands for center of mass by quite a bit. So you get this sort of higher order error if you want. And that's not good. So the question is, what can you do? And the answer is you can, rather than evaluating the center of mass of the whole green area here, you can just evaluate the center of mass of the pink area. This has the advantage that changes in the thresholding has only a very small influence at first on the center of mass. So that means you don't have these higher order errors, and of course you've sort of lost photons, if you want, for your center of mass, because that sort of pedestal is gone. However, you get a more accurate estimate. So I've done some simulation in my PhD that shows nicely that this is a much better way to obtain a localization than just based on the center of mass after thresholding and not changing the zero. So you can, in a way, you can say first subtract the background, clip at 0, and determine the center of mass of what is left. So here's some result from our lab, which is a STORM or dSTORM reconstruction, direct storm reconstruction of actin filaments that have been imaged by Ivana Shumanova-Chestak. And on the right-hand side, you see the wide field image as it is obtained by adding all these single molecular events. And when you then compare this to the image you obtain from the localization, so after taking something like 10 to 20 minutes of data, you can localize them, and you can actually then see and appreciate that this is a much higher resolution image. And what you also can see that it's not a problem to actually separate things if they're even 40 nanometer apart. And some really good data, for example, from the Sauer group, shows you that you can actually even separate you can see the hollowness of the microtubule when you antibody stain it, because the antibodies are sitting only on the outside. You can actually appreciate that these are hollow structures. And if you do three-dimensional localization, for example, by exploiting astigmatism or something like this, and reconstruct them in 3D, it's a really good test to see how good your resolution is by seeing, do we get a hollow microtubule structure? So the problem is, of course, that you can't be really fast because you need to acquire these single molecular images. At least that was the thinking. So interesting, Susan Cox in my group at King's College basically was going back to the idea of overlapping particles. So rather than having the particles completely separate so that you can then localize them individually, we go back to this original idea of having blinking and overlapping particles, but instead of performing an independent component analysis, what Susan and her collaborators were doing is to do a Bayesian analysis. So basically a maximum likelihood model that fits everything and basically looks at, it gets you something that I would call a probability map where possible molecule locations could have been. And then you're showing this to the user, this probability map. And the cool thing is that this can be done with molecules that are blinking in living tissue. And they are usually not as dark, so you will always have more overlapping images, but therefore you can be faster in the end. And so what you will see is some live cell data now that comes from, that has typically a frame to frame time of 6 seconds. So that means there were thousands of images taken in these 6 seconds, but they were localized with a sliding window and in a very computationally intensive way. But it shows you then a movie basically in living cells. So the model behind this is actually to make a hidden Markov model where the molecules can have several states, basically a non-emitting state, an emitting state, and a bleach state. And then even all these unknowns are sort of integrated out, and in the end you can reconstruct what you're interested in, which is this spatial probability map, so to say. So here you such a result. So you see the raw data like this, which looks at first very non-informative, but you might appreciate these blinking molecules. And even now there's still information in there. And if you just add all the images together, you see this photosome structure that was well known. So these are ring-like structures. However, if you now see what the algorithm can get from this data, it's actually quite amazing. It looks like this. So this is the probabilities where molecular positions could have been, and you can appreciate this very high level of detail. And that's even done or even possible to do in live cells. And the next video will show you a comparison of what I call pointillism, or 3B stands for Bayesian reconstruction of blinking and bleaching, or Bayesian modeling of blinking and bleaching. And so on the left side here, you can see a wide field series of images. So this is the sum of all the individual images that you can see here, and you can appreciate the resolution is by far not as good as the reconstruction obtained from the 3B method Bayesian modeling of blinking and bleaching, which you can see on the right-hand side here. So you can appreciate the very high resolution. The time between every frame is about 6 seconds in this movie, and the reconstruction took a very large amount of computer time because it is a very computationally expensive method. You can actually reconstruct this data on GPU, making it significantly faster, yet it is still a very computationally expensive method. So the problem there was that the reconstruction took, I think, something like a week for this movie on a computer. And that's, of course, a very computationally intensive method. There was some later publication by Susan's group that showed that you can speed this up with GPU processing. However, we also came up with another way, which is called non-negative matrix factorization, which is also a reasonably fast way to reconstruct blinking data. And here some information from that publication. So this was work by Andre Mandula, who worked on this in his PhD. And what this shows is a comparison of different methods. So this is the wide field overlap, of course, of a simulated data set on the top and an experimental data set below. So in the experimental data set, you can appreciate that these are probably two filaments crossing here. And the question is, what do the different algorithms give you? And you see that, for example, dSTORM as one of these algorithms seem to be having problems with these overlapping data where they cross. The same is true for bSOPHY, and 3B, the algorithm you've just seen, has also trouble where these filaments are crossing because it cannot quite figure out how to separate things, basically. But INMF, so the non-negative matrix factorization, and the I stands for iterative restarts, seems to be doing pretty well in actually reconstructing these particular situations. We have not had a really good data set to with overlapping samples to really process. What you see, well, this is INMF. This is with quantum dots, blinking quantum dots. Also, the other example with blinking quantum dots. You can see the filaments here that we can reconstruct of a cell there, which is also from the same publication. Right. So I want to mention that recently a method has been invented that's called Miniflux. And so the idea is to decouple yourself from the emitted photon picture. And so it's basically using a donut-shaped excitation and knowing as a prior knowledge that your fluorophore is active and there, and then the idea, if you place that fluorophore near the zero and if you're observing close to 0 photons, you know it is very close to the middle of the donut, and this way, without bleaching the molecule very much, you can actually find out that the molecule was very, very close to the center of that donut, as you see that here. So Stefan Hell's group, Balzarotti et al., they have done this fantastic technical implementation where very quickly you're going to 3 positions, so you're always sort of circling with your donuts, and by looking at the fluorescence response, you can learn in which direction your molecule has moved. And this way, you're actually able, without exposing the molecule to very much light, to track it with extremely high precision in the nanometer range over a fairly long time. And so this has been a fantastic application. And very recently, there was also a publication where they showed images using this method of Miniflux. Also here they showed some images of artificial origami structures, but in the new publication they showed also biological images. The problem with this method, since it's again a single point probing method, it's kind of very cumbersome to get enough imaging data out of it. Right. So I want to this presentation by simply showing you the same organelle structure, which are these podosomes, imaged in various modes. So you see a wide field image of podosomes, then you see a confocal image of podosomes of a cell. So podosomes are little vincular ring structures that cells use to dissolve bone, for example, certain types of cells. And this is a STED picture of protosomes. This is a structured illumination picture of protosomes. And this is what you've seen already, the 3B single molecule picture of these protosome structures. So the take-home message, well, before we come to the summary, some application notes. So structured illumination, linear structured illumination has the advantage that you can do many colors, and you're quite free in choice of dyes, and you can apply it to living cells. But of course, you get only a moderate resolution because it is a linear technique, just giving you a factor of 2 over wide field, basically. The same sort of arguments you can apply to other methods like the image inversion interferometry called UTSI, the ARIScan microscope, or ISM, image scanning microscopy, OPRA being an optical version of image scanning microscopy, also called photon, so optical photon reassignment microscopy. And yes, so then STED, being stimulated emission stimulated emission depletion microscopy, gets you a higher resolution. However, it works with very high intensities. Is a point scanning method typically. Of course, it can be also done with multiple points. It is challenging to do live cell imaging. It has been shown that we have some fantastic results. And it doesn't need image reconstruction, which of course is an advantage. Yet it can profit from it. Image scanning microscopy has, like I said, is pretty similar to SIM, so it's a high resolution, but not as good really as SIM, almost as good. But STED is clearly higher resolution. ISM is very light efficient, very easy to use, and because it's also beam scanning method, the errors are usually not as disastrous, if there are errors, as they can be sometimes in structured illumination microscopy. So the image reconstruction is trivial by just sort of joining the data in the right way. PALM and STORM, in a way, have the highest resolution, and of course Miniflux as well. And it can be done in multicolor. 3D is possible, however, not really easy. Live cell imaging, as you've seen, is hard, but in theory also possible, also in practice, as you see. So then I want to summarize. Many microscopic modes exist. Linear methods give you a factor of 2 in resolution. Nonlinear methods are unlimited in resolution. They basically circumvent the upper limit because of the nonlinear dependencies. With that, I want to thank lots of people, and a few of them are named here, that contributed somehow to the results that you've seen here. So Ronnie Förster, Jan Song, Aurélie Jost, Kozube Watanabe, Enno Oldewurtel, Andre Mandula, Susan Cox, Ivana Sumano-Cheskas, Kai Wicker, and of course all my group now and previous times. So thank you very much for listening, and then see you in the lecture discussion.